Hey everybody, it's Mr. Heisey. I'm here to talk to you about the immune system today. I'm actually down in my basement right now recording this at 10:11 in the morning, getting ready for you guys to hopefully be watching this video today later on in AP Bio. My kids are sleeping, so hopefully we won't be interrupted by any manic screaming or anything like that, but I'm going to do my best for you here. I'm going to try to get through the first 25 or 30 slides or so in the immune system slide so that we can wrap that up tomorrow. Of course, we'll also take questions on the problem set tomorrow because don't forget it is due on Thursday. So, Chapter 24 is all about the immune system. This is a surprisingly complex chapter. There's a lot going on and a lot of different types of cells to review and a lot of different interactions going on here, but they all have this theme in common. We are trying to get rid of the dangerous stuff, bacteria and viruses, and other invaders that can cause problems to our overall health. Let's start with a little bit of history, though. First, I want to mention to you somebody named Edward Jenner. This is an early doctor, actually, who noticed that women that were working around cows didn't contract smallpox at the same rate as the rest of the background population. Now, what this means is that these women were being exposed to the smallpox virus, presumably in a low enough dose that it wasn't something that was actively affecting them. They were basically working around these cows and forming some sort of sense of immunization to them because they were exposed to this virus. This is something that pops up a few different points in history where you can see that native populations really suffer with diseases because they have never been exposed to them before, and that's a big problem for them. So, it's interesting the way this science of immunology kind of begins just by noticing that people are not contracting disease the same way, and it has to do with their exposure in the environment to certain types of viruses or bacteria. It's also worth mentioning here the 1918 swine flu pandemic. This is right at the end of World War I. And you'll notice the number in the caption here, 22 million people killed in 18 months. An absolutely astounding number from a strain of flu. Flu continues to be something that's very dangerous today. There are actually thousands of people in the United States killed by the flu every single year. And of course, there are certain strains of it, like the swine flu in 1918, that do pop back up every so often, and they're surprisingly dangerous. This is this worldwide pandemic. A pandemic, by the way, is just another word for an epidemic that stretches across an entire continent. So it's not isolated to like a city or a population. It literally hits an entire portion of the world. And the swine flu pandemic actually spread all around the world. Every single continent was touched by this. Actually, very frightening stuff in 1918. So as we go through these slides, what I'm going to be focusing on here is on a couple different types of immunity. You'll notice we have innate immunity at the top of this slide. These are all things that are non-specific responses. They're something where you always have these systems working, like your skin is always blocking out viruses or bacteria. You've got phagocytic cells that are always going and trying to gobble up invaders, but they're not necessarily part of this specific response. So acquired immunity is something that works slower, but it's kind of like the targeted response of your immune system. This is something where you've got B cells and T cells. Remember, B cells are associated with the humoral response, creating antibodies that kind of flow around your body in the bodily fluids, and then you have the cell-mediated response associated with the T cells. These are the cells that specifically go around trying to get that one invader that you're dealing with right there. So innate immunity are kind of like the immune system's response that are always up. You've always got your skin protecting you. You've always got natural killer cells going around trying to get problem cells. Acquired immunity is the specific response. It's the clonally selected for cells, the ones that are actually going to end up doing most of the heavy lifting in terms of a specific problem, a specific invader into your system. So let's go over some different types of immune cells. There's a whole list of these white blood cells here. You can see that what's labeled as granular cells, all sorts of different jobs, all, though, again, with this same common theme of trying to get rid of bacteria and viruses and sometimes cancer cells that are causing you some sort of health problem. So we've got red blood cells up at the top. We know what they do at this point. That's more of a circulatory system thing. But when we look through this entire list of white blood cells, there's a couple that I want to point out to you that are things that are going to end up playing like a really important role in this stuff. Mast cells, about halfway down that list. You can see that they release histamine when they're damaged. Histamine, of course, is important in this like inflammatory response. This is also one of those things. It's one of these chemicals that tends to go wrong if you suffer from allergies. Hence why antihistamines help get rid of the symptoms of bad allergies. You'll notice the monocytes just below that develop into macrophages, which we'll be seeing. Macrophages then engulf and digest these microorganisms and activate T cells. There's all sorts of stuff that's going on here, okay? And then we have the lymphocytes, and these are the B cells and the T cells specifically. Notice the natural killer cells are part of these as well. Natural killer cells actually seem to be specialized towards getting rid of cells that are already infected or cancer cells. Your immune system actually plays an really important role in getting rid of tumors and cancers. It's actually very good at this, and this is a promising area of research right now in biology. There's a few different teams working on stimulating the immune system to go and attack certain types of cancers. Your immune system does this naturally, but their area of research is trying to get these cells to be more active in developing defenses against specific types of tumors. So if you come down with a certain form of cancer, the idea being you can basically sort of artificially order the natural killer cells to go and try to nab up and engulf and eat up the tumor. And actually, there's some really surprisingly strong results from this right now. There was just a study published about a month ago in a population of mice that they basically induced tumors into, and using this immune system stimulation technique, they were able to get rid of cancer in every single one of the test mice, which is really incredible. If you think about the implications of this, if we can go and refine this technique and make it safe for humans and spread this, throughout the medical world, then we basically solve cancer, which is kind of incredible. Not saying that we're there yet, but there is some really promising research stimulating those natural killer cells. So some very interesting stuff, and certainly lots of different types of cells to be thinking about here. So the non-specific defenses, remember, these are the ones that are always on, they're always operating. There's all sorts of stuff as you look down this list. Notice a few of them have to do with pH, creating acidic conditions. Others are simply like barriers, like your skin and your mucus. Okay, it just captures things and it keeps out pathogens. This is why you oftentimes have to worry about an infection with the cut. What you're doing is you're violating one of the non-specific defenses. So this can be a problem and an easy entry point for pathogens. Notice tears and saliva actually have got a specific enzyme called lysozyme, which destroys bacteria. So your tears and your saliva actually help to sanitize some of those moist surfaces of your body that could otherwise be open areas for infection. Lots of non-specific cellular, chemical and coordinated defenses, just kind of reading off the slide here. Coughing and sneezing, a fever is actually something your immune system intentionally does. You get a higher temperature because it increases the rate of reaction of these cells. It also is something that tends to cook out microbes that might be well adapted for the temperature range, the normal temperature range in your body. This, of course, can be a problem, though, because if your fever is too bad, then you end up going and denaturing your own liver enzymes and things like that, and you can actually pass away from that fever. So if you ever have a really bad fever, you end up going to the hospital for it. One of the things that they will try to do is cool your body down, simply so that you can keep your own enzymes in good shape. It's too much of a good thing in this case. So let's talk about the lymphatic system for a second or two. Now, we hit some of this yesterday when we were going through the circulatory system, because, of course, the lymphatic system is kind of like the gutter system is what we related it to, in that it returns excess fluid and leakage from capillary beds back into the bloodstream. But it also plays a really central role in immunity as well. These nodes that you see all over the body right here, which oftentimes swell up when you have an infection, these are places where B cells and T cells, those lymphocytes, they're going to be hanging out, waiting to be clonally selected for. So these are literally filled with all of these immune system cells just waiting to be selected so that they can do their job. Here's a lymphatic vessel. You'll notice this is structurally really similar to a vein in that it's got these valves. This is a one-directional system. Remember, there's no active pumping in this. Okay, there's no heart for the lymphatic system. So we can't rely just on fluid pressure. This is something where we are kind of relying on like body movement to be able to go and keep all of this excess fluid kind of passing through the lymphatic system. This is why people with really sedentary lifestyles, especially if they're a little bit older and they have circulation issues to begin with, one of the things that can happen to them is that water starts to build up in their, especially their legs is oftentimes where you see it. And they have like serious problems with swelling. And this can really inhibit their ability to move and be comfortable, even if they're just kind of like hanging around sitting in a chair or something. It can still be a real problem for them. And that is because there is no actual pumping method for the lymphatic system. You have to keep moving. It's skeletal muscle movement that actually causes these fluids to be moving through your body efficiently. So let's focus on a lymph node for a second or two, where these B cells and T cells are kind of clustered in there. So look carefully at the interior of this lymph node. You can see all those lymphocytes sitting in there, those B cells and T cells, literally waiting, being filtered through, sitting in the middle of all the lymph that's passing through this to see if they are going to be selected for. And we'll take a closer look at that actual process of clonal selection in a minute. Skin, of course, is a frontline defense for this. Notice how much is involved in your skin. Okay, we've got oil glands secreting this oil. That's something that's actually very bad for bacteria. Believe it or not, there are some cases where keeping your skin a little bit too clean can actually be a negative thing for you. It can cause skin to dry out and crack and actually violate some of the frontline defenses that the skin offers you. So there's a lot going on in this stuff, okay? But this is something that simply provides a barrier on the outside of your body so the bacteria and viruses can't easily enter into your system. Okay, you can see these white blood cells here starting to make their way through this little capillary. Notice it's squeezing through these cells here into what's labeled in the picture as the surrounding tissue, but basically the place that's being affected. You can see the bacteria right there getting engulfed by these white blood cells. These phagocytic leukocytes are mashing their way through the capillary bed so that they can go and respond to this. These cells actually do chase down bacteria. I've seen GIFs online of these white blood cells literally chasing bacteria around. The bacteria are running away and these cells trying to catch them. It's very impressive stuff, actually. But you can see they are leaving, in this case, the bloodstream to go into the surrounding tissue so that they can go and play a role in engulfing those bacteria and getting rid of the foreign invader. And this is part of the idea behind an inflammatory response as well. You'll notice these mast cells have left the bloodstream, okay, and they're going and starting to make this area swell, which goes and makes it easier for other cells to be able to get into this area. You can see the phagocytes in the second picture starting to gobble up some of the bacteria that were there because of this person's splinter. They're also releasing complement proteins. And those complement proteins are going to play an important role in lysing or breaking down the cell membranes of the bacteria. They also are going to cover the bacteria and basically mark it for destruction for the other phagocytes, so they can go and gobble those things up. Lots going on in this inflammatory response right here. Notice, by the way, in the third picture that we have these phagocytes, after they have gone and done their job, some of them will be damaged, some of them will be infected. Those things are gobbled up and digested, and the parts are recycled. Okay, here's an interesting picture, actually, of a white blood cell battling some bacteria. The bacteria, the little rod-shaped things, kind of look like little pieces of mouse poop in a way, or grains of rice, kind of covering this thing. But what it's doing is it's spreading out these little tendrils, and it's grabbing those bacteria, and it's sucking those bacteria in so they can be marked for destruction. Let's take a closer look at that process of going and pulling in those foreign bacteria. This is a process called phagocytosis. So notice what's happening at the top of the slide. Those little microbes, they're getting surrounded and basically placed into what's kind of like a vacuole. Okay, it forms from the outside. Those little pseudopodia that we've actually seen before in this class, engulfing the microbes, forming the membrane for the vacuole itself. And what this cell does then is it takes a lysosome. Now remember, a lysosome is basically a vacuole that's holding digestive enzymes. They get rid of garbage and broken down things in the cells. And it's going and just simply fusing the vacuole that contains the bacteria with the lysosome. The lysosome releases those digestive enzymes and it just breaks these things apart. Okay, the microbial debris you can see in the last step right there is being released in the process of exocytosis. So phagocytosis, this whole thing with the pseudopody are swallowing up bacteria. The vacuoles are joining with a lysosome. The lysosome and its enzymes are going and digesting the bacteria, and we have gotten rid of our problem at this point. Okay, so other frontline defenses. Interferon is a substance that is released by a cell that is already infected. So to be clear on this slide, the cell on the left-hand side is already dealing with an infection. It's not going to make it. But as it is in its death throes, the virus has gone and inserted its DNA, and as it turns out, the insertion of that DNA triggers the interferon genes to be turned on, and the cell, in its last dying gasps of this infection here, manufactures these interferon molecules, releases it into the bloodstream, and what it does then is it stimulates other cells nearby to go and start manufacturing antiviral proteins. So it is turning on other cells' genes by releasing this interferon stuff that makes those cells have a greater immunity to viral infection. So it's kind of like one cell in its death throes sounding the alarm for the other cells, and they start their defense mechanisms that allow them to be able to block the viruses. So this is something, by the way, that is specific with the viruses. The complement system. Complement proteins are these little proteins that can actually form together. Notice on the right-hand side it says this complement pore, and you've got this water flowing in. These things basically go and punch holes into bacteria. Now you're going and violating the membrane, and what this does is it allows you to be able to upset the osmotic balance of the bacteria, and it kills these things off. So these complement pores are literally little holes in the membrane that form that allow fluid to be able to pass through, either in or out, depending on the conditions around the bacteria. Notice the bacteria also being covered in these complement proteins, and this is something that is marking these bacteria for destructions. They are going to be swallowed up by the phagocyte because they are covered in these complement proteins. So very useful little proteins, a couple of different functions that they have. Here's a closer look at how these complement proteins actually violate the cell membrane of the bacteria. So you can see the plasma membrane here actually being kind of spanned by a complex of these complement proteins that literally just punch a hole in it. They form a hole and they lyse the bacterial cell. As it turns out, natural killer cells also use something similar to complement, a protein called perforin, and you could guess from the name what this does is it punches holes in things. It perforates them to help lyse these cancer cells that it's dealing with. Remember, one of the things that natural killer cells do is they hunt out these cancer cells. They also play a role in hunting out virally infected cells too, but it's really cancer cells is oftentimes the way we end up talking about NK cells. So what these things are doing is releasing these vesicles filled with perforin that are doing the exact same thing to the targeted cell, to the cancerous cell. Now remember, a cancerous cell is going to be a body cell. It's just a body cell kind of gone to the dark side. So these things are able to go and find these cancerous cells. They release these vesicles through endocytosis, and they get this perforin to go and punch these holes through. And again, this lyses the cell. It's something that makes it impossible for the cell to be able to go and survive. Now, the immune system has got to get a couple things right as it's working. One of the big functions the immune system has to do is it's got to be able to tell what's a foreign invader and what it isn't. And this is where these major histocompatibility complexes, or MHCs, actually play an important role. These are surface level proteins, basically sit in the membrane of the protein, and they act as like identification tags. It's basically your immune system's way to be able to say like, okay, that thing right there, that is a body cell. You have to keep that thing in place. I'm not going to touch that and mess with it unless it's got some other kind of like foreign invader going on. So I'm going to move elsewhere and keep looking around. So when a white blood cell comes up to like a normal cell in your body that is just doing its job and minding its own business and doing everything correctly, it doesn't react against it. Now, there's a whole host of diseases where this does not work in quite the same way. You know, when we talk about any kind of disease where your immune system is mounting an improper response, allergies can be a form of this, but it's not against your own body cells. The alopecia, that's the hair thing that I have, that's a form of this. It's an autoimmune disease. Type 1 diabetes is an autoimmune disease where your immune system actually attacks your pancreatic cells, and because of that, you are not able to go and produce insulin at normal levels. Rheumatoid arthritis is another form of autoimmune disease where the cartilage between your joints is literally attacked by these cells, and so people end up having really badly swollen joints. Lupus can be a potentially very serious form of autoimmune disease. There are definitely less dangerous forms of lupus, but sometimes you find people that are really greatly affected by it. So autoimmune diseases are more common than you would think, and they are certainly something to be aware of when we talk about the immune system. So let's talk for a second about antigenic determinants. I can't talk, sorry. This is the dangers of trying to do things like this. Antigenic determinants are these small little bits of proteins that have been discovered as part of a foreign invader. Now you'll notice here, it's the actual part that the antibody is reacting with. Okay, so an antigenic determinant is the portion that the antibody is mounting this attack towards. Let's take a closer look at the antibody itself. Okay, antibodies always have some variation of this Y shape, and it's important to point out that these are enormous proteins. Look at the picture at the bottom there to get a sense of just how big these things actually are. And it's really a complex of a couple different proteins all working together. There's something called the constant region, which forms like the backbone, the Y shape of the antibody, and then you have the variable regions. The variable regions, well, they're not the same from one antibody to the next, unless you have something that's being clonally selected for, and then you're manufacturing one specific type of antibody as part of a humoral response. So these variable regions up here, these have got active sites, and they're actually something that's just kind of randomly produced. There's lots and lots of variation in how these things are made. And the idea behind that is that when you get an antigen somewhere floating around in the bloodstream, some foreign invader, it is likely that you might have something already in you because of this huge variety of antibodies that your body has created that is going to react against that. And if it does react against that, if the variable region in one of these antibodies has done its job, then at that point, we've got an antigen detected, and we can go and start the process of clonal selection. So you might be wondering, how do we make all these different types of antibodies then? You know, if we've got these millions of different antibodies literally floating around, in our lymphatic system, then where do these things really come from? Well, as it turns out, it's actually kind of a random mixing of DNA. You might remember this looks a bit like some of the slides that we looked at earlier in this class when we're talking about genetic regulation and discussing how you can go and mix up the order of exons in a strand of DNA, or excuse me, a strand of RNA to be able to go and give you a variety of proteins even from just one genetic sequence. We have something similar going on here that's making the variable regions. Okay, so the constant region is always made. It is the same from antibody to antibody. The variable regions, which contain the active sites, those are not always the same. And because of this exon shuffling that the mRNA strand can do in its trimming stages, then we can have a huge variety of these antibodies floating around ready to go and help stimulate a response. Here's a few different types of antibodies. This is a lot of detail for what the test might ask, so I wouldn't worry too much about this, but I do want you to notice that these are all variations of the same Y shape, just put together in different ways. So let's start diving then into the humoral versus cell-mediated immune responses. So we're getting into the things that are going to be clonally selected here for. So let's review this really quickly. You might remember the B cells are the ones involved in humoral immunity. Those are literally going to be the antibody creating cells. Those are the ones that are making the specific version of the antibody that is reacting against a specific and foreign invader that's giving you a problem right now. Now, this does all of its maturing in the bone marrow when we talk about these B cells. You'll notice here the thymus actually plays an important role, though, in helping the development of these T cells. So while the stem cells for these things and the lymphocytes are actually manufactured in bone marrow, they then transfer in the bloodstream to the thymus, and they literally wait there and mature into like a full-grown T cell. So there's automatically like a maturity difference in the way these things come to be into their adult forms between the B cells and T cells. At that point, once they are mature, then they're traveling into the lymphatic system, and these things are hanging out in lymph nodes waiting to be clonally selected for. So let's look at this process of actually mounting an immune response in the specific systems. Okay, what we have here is a little bacteria being swallowed up here, phagocytosis, remember, in this macrophage, and it's being broken apart. So as it's being broken apart, the cell actually does something kind of clever. It literally takes the pieces of the bacteria, and remember, those pieces of bacteria are going to have some of these identifying proteins sitting on them, and it goes and basically sticks them out to the outside of its body, and it takes on the role of an antigen-presenting cell. So what's happening here is these little bits of protein and membrane from the bacteria that mark it as what it is are being put on the outside of the body. Remember, those little bits of proteins are called antigenic determinants. So the antigenic determinant is being held out on the outside of the cell, and it's saying, hey, look, I've found something. And then from that point on, you can see this helper T cell is going and interacting with this thing. So the antigen-presenting cell is presenting the antigenic determinant, which is really just a piece of a bacteria or a virus that it found, and it's going and showing that thing essentially to a helper T cell. The helper T cell then is going to be responsible for going and stimulating the rest of the clonal selection response. So notice what is happening then. We've got this activation phase going on. The antigen-presenting cell, the macrophage, is going and holding this thing out. Now, you'll notice it's starting to also secrete this stuff called interleukin-1. Interleukin-1 is helping to activate the helper T cell. So the macrophage is sounding the alarm. It's saying, hey, I found something. Look at this. It's putting out this chemical that goes and stimulates the helper T cells. The helper T cells then respond by going and releasing this stuff called these cytokines here. Now, this substance that the helper T cells are secreting, you can see it like midway through the slide, halfway up in the image. This is actually something that's going to go and stimulate the helper T cells to go and clone themselves. So we're making a whole lot of these helper T cells now, ones that have seen that antigen and been stimulated by it. So these helper T cells then, they go and they interact with these B cells. You can see this is called the effector phase in this slide. Now what's happening here is that the helper T cell has gone and taken this from the antigen presenting cell. This antigenic fragment, this little bit of bacteria, then is going and interacting with these B cells and stimulating the correct B cells. Remember, there are lots of different varieties of these things because they all have these different antibodies. So it finds a B cell with the correct antibodies in the active site and the variable region of its antibodies that they can actually respond to whatever it's got. And when it has a match, when it actually finds a B cell that is going to be useful here, it releases lots of this cytokines again. And this, remember, is the substance that causes these immune system cells to start multiplying. So once we find the correct B cell at that point, then it's a matter of going and making more of them. So lots and lots of B cells are made. This does take time. It takes a couple days, which is why you feel sick and gross for a little while you have an infection before your immune system starts to really kind of kick into gear. Remember, you've got to make a lot of these cells. And so this is something that does simply take some time. Now, some of those cells are automatically going to be stored from the very beginning off to the side. You're going to keep those things as memory cells. The reason why these memory cells are important is because if you come across an antigen once in the environment, you are likely to come across it again. Okay, so if you can go and keep some of these memory cells that have already been clonally selected for in waiting, then it decreases this amount of time that you can have before you have another specific immune response should you get infected a second time. The rest of the cells, the B cells that are not being held in reserve as memory cells, basically turn into plasma cells that are going to start cranking out lots and lots and lots of these antibodies. As you might imagine, these B cells have lots of rough endoplasmic reticulum, which makes sense because remember, rough endoplasmic reticulum's job is to go and manufacture proteins. These antibodies are proteins. So these B cells have got truckloads of this rough endoplasmic reticulum because they are antibody factories. They make lots of this stuff. Here's an interesting plasma cell. You can see all the rough endoplasmic reticulum taking up nearly all the space in the cytoplasm here. These things are doing their job, creating as many antibodies as quickly as possible. So what do these antibodies do as they're floating around in the bloodstream then? Well, they go, and remember, these are antibodies that were specifically chosen because they can bind with the antigenic determinants, which happen to be covering all of the other bacteria or viruses that's giving you problems with this infection. So the antibodies are able to go and basically, with their active sites in the variable region, kind of bind themselves or glue themselves to the outside of the bacteria or the viruses that are causing this infection, to the outside of the antigens, in other words, and they mark it for destruction. It's kind of like one of those like bags of money that you see in the news sometimes, where, like, you know, the cops will hand over a bag of money to somebody that's holding some hostages, and the bag of money blows up and it's like filled with dye, and the person is just like, you know, covered in purple paint or something like that. You know then who's the guilty party. You know, even if they run out and they cause problems, you can always find them again because they're marked. They're marked for destruction. In this case, those bacteria are marked for destruction by the antibodies. So this is a really important function of these antibodies in the bloodstream. The antibodies are also capable of actually getting these bacteria to sort of clump together as well, which make it easier for phagocytes to go and swallow them up. And these antibodies also play an important role in activating the complement system. Remember, these complement proteins are the ones that punch these little holes. They force these pores to open up in these cells, and they kill off the infected cells, or the foreign cells, rather. So you might be wondering, you know, how do the complement proteins know which cells to join into? Do they just randomly go around? They don't, okay? They are actually stimulated by the presence of antibodies that have gone and bound to the antigenic determinants on the outside of the foreign invading cells. So the complement system is actually a targeted system, but it's not targeted specifically itself to the antigenic determinants. It uses antibodies to go and be stimulated so that it can mount its own kind of defense and forming these little pores and causing all the problems for the foreign invading cells. So these complement proteins, again, coming in the mix here and causing problems. All right, so folks, I'm about 31 slides in. That's actually where I wanted to be for the day here. We're going to take care of our last 15 or so slides tomorrow and look at the rest of the specific immune response with the T cells. So either way, I hope this worked okay for you. I'm sorry to not be there with you today. Hopefully we'll get all this stuff taken care of, and I will be back with you tomorrow. I will see you later, guys.